Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 179th seminar, and I call it a J. Craig Venter Institute Day. Science and technology often advance with a breakthrough moment, which occurs thanks to pioneers or visionary researchers. Recently, unfortunately, we lost two such pioneers at J. Craig Venter Institute, including Professor Clyde Hutchinson, a member of National Academy of Science, and Professor Ham Smith, a Nobel laureate, both leading early sequencing efforts, including the first complete sequence of a DNA molecule and also the human genome, as well as building cells containing the synthetic genome or the first synthetic genome. Although they pass away, their legacy will remain with us as pioneers who studied early molecular biology and synthetic cell research. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Chris Dupont. He is a full professor at J. Craig Venter Institute, where I also working at. One of the oldest members leading the institute for a while. In my opinion, despite our recent loss, Chris and others will continue to demonstrate our excellence in pioneering research. Chris also doing a lot of sequencing work, also bioinformatics as well. It is my great honor to work with him on some project, including our NSF Global Center project, where he is in charge of the bioprospecting thrust to discover new enzymes and strains for waste valorization. Chris, thanks so much for your leadership and also contribution to the scientific society. The virtual podium is all yours now, and thank you again.
Thank you very much, Taesuk. It's really appropriate that you mentioned Clyde and Ham at the beginning of this. So I don't have any slides today, but I wanted to talk about how we are approaching a convergence of technologies that is really going to open up a lot of opportunities in synthetic biology and science writ large. And it ties back to some of the work that Clyde and Ham were involved with. One of Clyde's biggest contributions to the Human Genome Project, as well as the first metagenomic sequencing of seawater, was building vectors that allowed us to do very accurate mate pair sequencing. We knew there were two Sanger reads 10 kb apart, and we thus had that informational position. And this led to, closing the human genome and essentially you sequencing the phase diploid human genome in 2007. Unfortunately, in genomic science, we kind of lost our way for a bit there while synthetic biology continued to push on and develop the tools to build chromosomes, bacterial and eukaryotic. The bulk of our genome sequencing, driven by human genome sequencing, was SNP capture or short read profiling, with no structural resolution of those genomes. And this has set human genomics back 15, 20 years. But it also meant that the huge diversity of microbial eukaryotes in the world, fungi included, were essentially profiled with no intent for structural resolution of their genomes. And this has really set us back. And I feel that in the next few years, we are going to see a wellspring of discoveries in chromosome biology that will open up incredible new tools in synthetic biology. For example, we already know that dinoflagellates, who have genomes bigger than human genomes, and they're mosaic composites of multiple ancestral endosymbiotic events, they sometimes will have little mini circular chromosomes. How do they do this? This is an incredible tool set. We know some Candida species can spontaneously alter ploidy of entire chromosome, their entire genome, but also individual chromosomes. Single chromosomes will suddenly become variable ploidy. And I anticipate as new accurate long read sequencing technologies that are driving a new form of human genome sequencing, they're also going to inform protist and eukaryotic genome sequencing. And we are going to discover incredible new facets of chromosome biology that will drive forward synthetic biology in the future. And these nature inspired solutions make me really, really excited despite the headwinds against science currently. And that is it.
Fantastic. So that is basically we are talking about very exciting future, even though first human genome sequencing is done uh, more than 2 decades ago, but still, new technology, regarding sequencing was to kind of make another breakthrough. And I believe the Chris, you're gonna do another, big breakthrough in near future. In that way, we benefit, synthetic biologists basically benefit from those kind of new breakthrough. So thank you so much for your wonderful contribution again. And I'm really, honored to be with you, doing a lot of different, project together, more project together in the future. Thank you.
A- Absolutely.
Fantastic. All right. Now, the main speaker of today with a longer introduction, Dr. Michael Cory is a synthetic biology Hive fellow in the Department of Systems Biology and at Harvard Medical School. He received his PhD in biochemistry and molecular biophysics at the University of Pennsylvania in the lab of Rahul Kohli, where he studied the molecular interface between LAG-A and LAX-A proteins governing the SOS response in bacteria. Through his PhD work, he became more interested in how bacteria respond on a molecular level to subtle environmental cues and how this biology could be leveraged in synthetic biology to perform a variety of industrially important tests. This burgeoning interest led him to the labs of Pam Silver and Michael Springer at Harvard for his postdoctoral research, where he is working on engineering non-model microbes for the sustainable extraction of rare earth elements from low-grade inputs like mining waste. Michael, thanks so much for your time today, and please take it away.
Thank you so much for the introduction. And I would like to also thank you for keeping this series going and organizing it. It's a pleasure to be able to finally take part. So today, let's get the slides up. So today, I'll be talking about biomining as a sustainable approach to rare earth purification. And while biomining is the backdrop, I think Chris did a good job of setting the stage in how we think about synthetic biology, whereas typically we fall into the trap of thinking like engineers. I argue that we need to take nature solutions and try to integrate them more into our work by sampling from the environment. And sequencing, now being cheaper than ever, enables us to do that. So before I get into the biomining aspect, I have to start with kind of a more downer topic, and that's climate change. Every ton of CO2 we put in the air matters, Here's from the previous IPCC report in 2023 showing that as we have cumulative CO2 in the atmosphere, we see a likewise warming of the surface temperature. Now you can project this forward to additional carbon we put into the air and calculate how much carbon can we put in before we reach, say, 1.5 degrees of warming. If you've been paying attention to the news recently, it seems like we may be already there. And in fact, if we continue our carbon emissions as if nothing's changing, through 2030, we'll blow past that. And we certainly aren't doing enough to change that. Now, there is some light at the end of the tunnel here, in that green technologies are becoming much cheaper and therefore much more widely adopted. So green technologies are just technologies that can generate energy without the need for putting more carbon in the air. So for example, solar voltaics, so solar energy, now is cost competitive with fossil fuels, and likewise, the market adoption is very high. This is a little bit of a paradox, though, that green technologies are actually driving the demand for kind of unsustainable materials. So I'm going to focus on the rare earth elements, which you may have also seen in the news recently. Out to 2050, the demand for these elements is projected to increase dramatically. And a lot of this is due to these electric vehicles and wind turbines. So the mining requirements are quite steep as well. Currently, in the yellow bars, we're producing as much rare earth as we kind of need to make all the technologies we use every day. But by 2040, regardless of which climate scenario you're looking at, either the announced pledges or the net zero, we aren't producing enough rare earths to meet the demand for those technologies. And this is a problem because producing rare earth elements is very, very cost intensive, very energy intensive, and produces a ton of really nasty pollutants. And the grade inputs you have to work with are much lower than things you would otherwise think of for mining, like copper. So 8% rare earth by weight rare earth element is considered, kind of higher grade input. So what are the rare earth elements and why are they so costly to mine and produce? So I've reconstructed the periodic table here to put the F block elements back where they belong. The rare earth elements are the first row of these F block elements, the lanthanides, plus scandium and yttrium. So it's not actually an abundance problem. The rare earths aren't all that rare. So here is the average abundance in North American shale compared to copper. And you can see the light rare earths are more abundant than copper even, with the heavies not being too much lower. So they're not really rare. Why are they hard to mine? Well, there's two problems. One is they're chemically cohesive as a group of elements. When you talk about transition elements like iron, copper, zinc, there's different oxidation states. They have very different chemistries, and thus you can utilize those properties to purify them. The rare earths are all for under most normal conditions +3 charge. They all behave chemically very similarly. The only real difference is as you go left to right across the periodic table, you see a slightly larger than average contraction of the ionic radius. And this is what people use as a basis for purifying, usually through chemical extractions. So because of these differences being so subtle, you need hundreds to thousands of steps to separate them from each other, which is compounded by the second problem. They all co-occur, and they're kind of sparsely distributed in the crust. They don't occur as very high-grade ores. So here's the cost per gram of these rare earth elements. And you can see you reach a cutoff point where once you start to get more rare, it's harder and harder to purify them from the more common ones. And that drives the price up. So terbium is very expensive, and it's used in high performance permanent magnets. Now, I've also highlighted here in green, we actually discovered that microbes can use these elements in their metabolism. In red, we have yet to observe that in nature, that organisms can use these elements. And unfortunately, that means the split is even worse with these red ones over here. So how do we turn an engineering problem of mining and purifying these elements to a biological one? How can we use microorganisms to purify instead of these costly chemical extractions? So typically when you mine, you have this starting ore that's a matrix of your target metal and then either other metals or other minerals like phosphate. And you got to break this apart. Industrially, this is done with heat, acid, toxic solvents. And then you need to beneficiate or enrich for your target metal, and then finally purify. So where does biology slot into this process? Well, biology is very, very good at getting trace elements from the environment that they need to grow and live. So you can either have an organism that needs your target metal or needs the other metals or other minerals in the matrix, and that can speed up the dissolution. And this is called bioleaching. So you can use microbes for either the leaching process or the beneficiation process, or you can take the peptides and proteins or small molecules that they produce to target these metals and use them as the basis of a purification. This gives us a few opportunities for synthetic biology to beef these organisms up. You can increase their capacity for storing these metals. You can increase their specificity for certain metals over others, or you can increase their leaching more broadly. So there's some benefits of biomining over traditional mining. Microbes can utilize much lower grade inputs. Evolution has tuned them to be looking for trace elements, so they're very good at trace input materials. This would be something like e-waste or mining waste, where the bulk of the metals have been removed. There's far fewer amounts of harsh chemicals needed to recover from cells. They're easy to lyse with non-chemical methods, and it's more energetically efficient depending on the metabolic requirements and the dry cell weight recovery of your microbe of interest. So in order to perform a function like biomining, you need to understand a lot about the biology of how those metals are trafficked and stored. And unfortunately, we're kind of limited because this is a newer field on how organisms use rare earth elements. Most of the work has been done in this model organism, Methylarubrum extorquens. It's a soil microbe. And the way it targets these metals for import is they produce small molecule chelators called lanthanophores that go out into the environment, target rare earth elements, transport into the periplasm. In the periplasm, that's where the metal's actually needed. The enzymes that they use for the metabolism is a methanol dehydrogenase, which I'll get to in a bit later. But it can also import into the cytosol for storage. And largely this is done through these polyphosphate granules, where polyphosphate kinase produces these polyphosphate molecules, which can form tight minerals with rare earth elements. There also may be other proteins involved in the storage that we have yet to discover. So we have a microbe with a desired function. That's the extraction and fractionation of rare earth elements from mining waste, for example. So here are the pillars we need to get there. One nice thing to have would be a guided evolution system of model systems. So this is kind of a more hands-off approach. If we can have a growth-based selection, then we can just let the evolution take its course. Second, we can do our synthetic biology thing, and that's the rational engineering of known components, either changing the proteins themselves or changing expression levels or gene circuits. And then finally, and this is, I think, the more exciting area that people overlook, is the discovery of new systems and microbes, looking in the environment to see what nature has already come up with as solutions. And to do each of these, those require certain things to be optimized in the lab. So you need, facile cloning toolkits, you need a method of survival-based selection, you need some solid bioinformatics pipeline to generate lead molecules that you can target, and you need a way to sample microbes from the environment. So the overall goal of my project is, can we learn more about lanthanide biology or find better engineerable chassis simply by sampling from the environment? And the way this pipeline works, for those unfamiliar, is you go out, you find an environment that's likely to have the properties you want. Since our model comes from the soil near roots of plants, let's do that. You inoculate into your selection media, do serial passaging, along with plating, colony isolation, and outgrowth, until hopefully you can get isolated microbes. Rather than, you can get communities, but they're a little bit harder to work with. So here's an example of 3 of the strains we've been able to get from our environmental isolation. It's really nice to see after a couple months of plating, picking, you finally get pure cultures. With genome sequencing being ever so cheap, we can sequence everything, characterize them, and we managed to get, over 12 different microbes from the environment, 6 of which are novel species. So we've isolated microbes. What do we do with them? First, we need to ask the question, do these microbes have similar systems to what we already understand for lanthanide biology? Second, can we do functional omics to better understand the lanthanome dark matter? things that we don't yet know that are important for lanthanide trafficking, storage, or sensing. And then third, application, application, application. Can any of these strains serve as better models for rare earth biomining, or how can we start engineering them to do the job better? So let's start with bioinformatics. Do these microbes have similar systems to what we already know and understand for lanthanide acquisition? So we have two good starting points based on all of the model work that's been done to date. One is the enzyme that uses rare earths as a cofactor, and that's this methanol dehydrogenase. And the second is a lanthanide binding protein called lanmodulin. So let's start with the dehydrogenase. In amextorkins, you can have two different flavors of these methanol dehydrogenases. One is XOXF. This is a lanthanide-dependent enzyme with a PQQ cofactor. This is the coordination environment. And you can have MXAF, or a calcium-dependent version of this enzyme. Now you'll notice they look very similar in their metal binding site, with the exception of XOXF forms a 10-coordinate ligation with the metal. The calcium-dependent is 8-coordinate. And the metal binding motif for XOXF is ENDD, for calcium, ENDA. And it turns out this residue right here is the greatest predictor of the metal use of these class of enzymes. If it uses calcium, it's going to be an A, S, or T. If it uses lanthanides, it's going to be a D. And this is the case for all the XOXF enzymes that have been looked at so far biochemically. As a proof of principle here, you can look at P. putida. This is a common industrial microbe and also a lanthanide user. It has PET-H, a lanthanide-dependent ethanol dehydrogenase, and PET-E, a calcium-dependent version. As you can see, pretty much the same principle applies. ENDD, 10-coordinate, lanthanide user. ENDS, 8.5-coordinate, calcium user. Again, this residue is very important. So we looked at all of the sequences in our environmental isolates for all of their different PQQ-dependent dehydrogenases. In addition, we threw in a bunch of known XOXF and MXAF members to build this phylogenetic tree where we aligned everything. And we can start to look at kind of the broader tertiary structure of these proteins. Here's your standard flavor PQQ-dependent dehydrogenase. You see this beta propeller fold. The PQQ in metal is in the center. And this is kind of our baseline. This group right here, they have this extra adapter domain that has a couple alpha helices. We don't really know what it's doing, but otherwise it looks very similar. Same here, where our alcohol dehydrogenases look very similar to the XOXF proteins. Similar here. This one is a kind of special group. These are glucose dehydrogenases, and they actually tether to the periplasmic membrane with this transmembrane domain. And this will be kind of important later. So now we can look at the metal coordination sites of each of these proteins and ask, is it probably lanthanide dependent or probably calcium dependent? And how does it cluster with the known families of these enzymes? So here we have these XOXF families that have been characterized before, and D, 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 all of these known lanthanide-dependent enzymes end in D, all the known calcium-dependent families end in S. Where we start to get deviation is over here in these glucose dehydrogenases. And I'm going to point specifically to this E. coli glucose dehydrogenase. Not a lot of biochemical work has been done on these enzymes, except for the E. coli one, and it is magnesium calcium dependent. You'll notice it does have a D here, but now they've lost this asparagine and replaced it with a valine. So the question is, are these groups that now have lanthanide-like active sites actually lanthanide-dependent glucose dehydrogenases? Is this a new family of lanthanide-dependent dehydrogenases that act on things beyond alcohols? So there's some conditional evidence here that might support this. A group recently published a paper looking at surface C metagenomics, transcriptomics, and they were asking the question, how does XOXF expression vary in different regions and across different timescales? And they found that XOXF cycles across day-night. And this coincides with methanol production by phytoplankton and other photosynthetic organisms. So as there's more methanol in the environment, they upregulate their lanthanide-dependent dehydrogenases to compensate. What's interesting is they also have these glucose dehydrogenase enzymes that are potentially lanthanide-dependent, And these coincide with the expression of these lanthanide-dependent methanol dehydrogenases. So ocean communities may be also tailoring their glucose dehydrogenase to coincide with the availability of methanol for some reason. Now that's important because these glucose dehydrogenases, largely what they're doing is producing this gluconic acid, which, as we know from the chemistry labs, can be used as pretty good chelators for rare earth elements. And organic acids can also liberate phosphate from the environment. And because rare earth elements form phosphate minerals, that could be kind of a two-step way to get more rare earths. Liberate it from the phosphate and chelate it. So we can bin all of our environmental isolates based on the presence of these calcium or lanthanide-dependent enzymes. As you would expect, most of them have both the calcium and lanthanide-dependent versions. However, we have one strain that has only the calcium-dependent version, and then one strain that only has the lanthanide-dependent version. This strain is very special because it can only grow on methanol when you give it rare earths in the media. And I think this is a special point to highlight because not a lot of people do environmental isolations where they think to put rare earths in their media. So if you're doing that, you might as well throw rare earth elements in. You might get something fun. And we can test these strains based on the expected phenotype on the presence of these different enzymes. And we can see this methylotenera only grows on methanol when you give it rare earths. When you don't, it doesn't grow at all. This Pseudomonas species that has both calcium and lanthanide-dependent versions can grow just fine whether or not you give it rare earth. So moving on to the next protein, we have lanmodulin. This is a rare earth binding protein, named such because it looks a lot like calmodulin. It has these EF hand motifs, which coordinate typically calcium, separated by these alpha helices. So it also has an 8 to 10 coordinate metal binding center. And the calmodulin EF hands, the primary difference is this position here is usually a threonine or a serine. In the lanmodulin or lanthanide binding peptides, these are usually aspartic acid. And work from the Curtruvo lab has been pretty critical in defining the proposed criteria for what makes a modulin a calmodulin or a lanmodulin. And that's largely the EF hands in lanmodulins are separated by 12 to 14 residues in these helices, whereas the calcium-dependent ones are typically 25 residues. There's usually about 4 EF hands in the protein. There's usually a proline at position 2 in the EF hand, and there's usually a carboxylate residue at position 9, typically aspartic acid. So how do our environmental isolates compare to the lanmodulin calmodulins, or do they have any? So methylotenera, which I established is pretty tailored to using rare earths in its metabolism, does have some EF hand proteins, but this one has the right spacing. One of the EF hands has the aspartic acid, but there's only 2 EF hands. Similarly, this Rosiobacter, which we didn't isolate, but we're also working with, it has, again, the lanmodulin spacing, but now this residue position is more calmodulin-like. And then finally, this A. methanolica, another strain we're working with, it has the right spacing, it has this aspartic acid at the right position, most of the EF hands, but it doesn't have any prolines in the second position. So unfortunately, it doesn't look like the established criterion seems to work all that globally as we would have liked. So now we're working on actually purifying all of these EF hand proteins and testing them in vitro to see, is there a better established criteria we can use? So the next steps for the bioinformatics, we've identified a potentially new family of lanthanide-dependent dehydrogenases that maybe use different substrates like glucose. Are they truly lanthanide-dependent? So we're working on purifying them and testing them in vitro and doing some knockouts to test that. And do these EF hand proteins or Pepsi proteins, which I'll get to in a second, do they bind lanthanides? So next, functional omics. So we've exhausted our limited resources in what we know about the lanthanome. How can we do some experiments to uncover new biology? It's a pretty simple experiment. We're going to do a transcriptomic experiment with our methylotenera and a couple of the other strains. We can grow it on methylamine and methanol. So our methylotenera can grow on methanol only when there's rare earths. It can grow on methylamine whether or not we give it rare earths. And we split it into different test cultures, one where we don't have any lanthanides at all, and one where we have a mixture of different lanthanides. And this is what the transcriptomics for methylotennero looks like. Over 50% of the annotated coding sequences in the strain are differentially regulated. Not surprising, it seems to have a pretty tightly, intimately coordinated metabolism with rare earth availability. So we can look at, some of the upregulated hits. So most of the upregulated hits are kind of various incidental metabolism changes. For example, we have this group of genes that are mainly for respiratory redox cycling. That makes sense. But we also have this novel CAR-RS type 2 component system. So CAR-RS is typically a calcium sensor and it was identified in Pseudomonas. This has a very similar architecture with these added Pepsi domain containing proteins. That's highly upregulated in the plus lanthanide condition. We also have these orphan EF hand proteins, one of which I have highlighted earlier, and that would be this one. And we also see an orphan Pepsi domain containing protein. Now, the downregulated genes, again, mostly incidental biology that may or may not be directly related to rare earths, though we do see a huge downregulation in these methylamine utilization proteins. That makes sense when it has both methanol and methylamine around. If it can't use the methanol because there's no rare earths, it's going to preferentially use the methylamine utilization genes. So let's talk a little bit about this two-component system. So the way this is architectured in our strains is we have these two Pepsi domain proteins. Sometimes they're in this inverted orientation to the operon, sometimes they're together. We have a response regulator, which is the operator binding DNA binding protein, and then a histidine kinase. And they're dictated by this upstream operator with a consensus site of TTAAG or TTCAG. And these Pepsi domain proteins are very interesting because they can form homo or heterodimers that form these cages of aspartic acids, which, if you'll recall, are very important for coordination of these rare earth elements. And as it turns out, while we were working on this work, another group was doing transcriptomics on a rare earth user, and they identified that one of these Pepsi domain proteins can bind very tightly and very specifically to rare earth elements. Now, another paper just came out about a week ago that took this protein, put it on a column, and for the first time ever, we were able to get separation of almost all of the lanthanides in a single step, single stage citrate elution gradient. So they were able to purify to pretty decently high purity each of the rare earths from a mixture by utilizing the very slight binding differences of this Pepsi protein for the different coordination environments of the different rare earths. So this is really exciting. So a lot of groups are now putting more effort into these Pepsi domain proteins. We found them ourselves in most of our environmentals. Here's how we think, or here's how generally the histidine kinase system works. You have this histidine kinase sensor that crosses the periplasm and cytoplasmic barrier. In the periplasm, there's a sensor domain that when there's a signal, it autophosphorylates. This phosphorylation can transfer over to the response regulator. And once phosphorylated, this regulator dimerizes and can bind to the operator sequences, leading to either upregulation, downregulation, or both, depending on the context. And as it turns out, almost every last one of our environmental species has this two-component system complete with the Pepsi domain proteins. And when there is this module, there's also an orphan Pepsi domain controlled by a similar operator elsewhere in the genome. So what we think is happening is this is a lanthanide sensor module two-component system, not dissimilar from the two-component system that's been identified in Pseudomonas putida that works to sense rare earth elements. And using a little bit of AlphaFold, playing around with different ratios of these operon components, what we think is happening is the histidine kinase dimerizes, which can bind to our regulator, which transfers a phosphate. This may or may not be mediated by a lanthanide ion or a calcium ion. And then the Pepsi domain proteins form a cap on the sensor domain and actually act to sense this calcium or lanthanide, depending on the system. And we can look at the predicted alignment error to kind of get a sense for how good this AlphaFold model might be. You can see there's a lot of variation, but largely the domains that are on the same side of the membrane tend to be decently high confidence. So moving on from this, this is very exciting ongoing work. We're working on some functional genomics to ask the question, is this truly a lanthanide sensor? How can we utilize this either as a sensor tool or to engineer the biology of these organisms? Now moving to the kind of hands-off evolution experiment. I mentioned this methylotenera requires rare earth elements to grow on methanol. So what we can do is we can passage it in a permissive rare earth, one it can use, and then passage it into a rare earth it can't use and see if it can adapt to now being able to use the heavier and heavier rare earths. And again, the cutoff for biology currently is this gadolinium to terbium cutoff. Now, I'm not going to put the cart before the horse here. This is ongoing work. We have some very exciting preliminary results. Hopefully, I will be able to talk about this down the road. If you go to the SynBYSS conference in Barcelona, I'm going to try to be there for this to talk about this work. So our next steps for the functional omics are these highly conserved two-component systems for lanthanide sensing? Did we find a lanthanide sensor that's more direct than the systems that have already been figured out? And how are some of these other seemingly unrelated gene clusters related to lanthanide homeostasis? Is it incidental metabolism related to other properties, or is it directly related to this lanthanide property, sensing property? And then finally, application. Can any of these strains serve as better models for rare earth biomining? So we did a kind of simple experiment where I collected a bunch of old phones and computers, discarded all the plastic, got all of the printed circuit boards and rare earth containing components like screens and permanent magnets, ground them up, and we're trying to answer two questions. Are the non-target metals toxic to cellular growth? And second, can we recover any rare earths from the waste? And our methylotenera is a perfect model for this because we know it's collecting rare earths from the waste if we see growth. So the way I set up this experiment is you have a solid input trapped in an agar matrix. You overlay it with a culture of your microbe of interest and see if it grows. So the methylotenera, when I don't add anything to the agar, it doesn't grow because there's no rare earths around. If I give it e-waste, it grows a little bit, but not too well. If I give it a ground up magnet, which we know is higher content of rare earths than general e-waste, it grows pretty well. And if I give it a ore simulant of xenotime, which is a rare earth containing ore, it can also grow. So it has a way to extract the rare earths from the solid matrix. If we give it supplemental neodymium in the media, then all of a sudden we see growth with no additives, and we see less growth with the e-waste. So what this implies to us is that the e-waste is probably toxic. There are other metals in there. Copper, silver are the big antimicrobial metals that might be posing an issue. But the key takeaways is we see strong growth on higher quality inputs, so ore simulant or recovered magnets, weak growth on general non-sorted e-waste, and e-waste may be posing toxicity risks at or above 5% pulp volume, which is what we used in this experiment. So our next steps for this application, how well do these strains biomine from a variety of input sources? So we can quantify this with ICP-MS, how much is in the cellular fraction, how much do they leach into the media, and can we begin engineering them for better biomining properties based on what we've learned about the lanthanide biology in step 1 and step 2? And as a broader summary, we have more work to do on the basic bioinformatics, more work to do in the functional omics, and our final kind of exciting application. So many thanks to members of the Springer Lab and the SynBio Hive. Particular thanks to Amog, Max, Matt, and Nathan, who have been kind of key to pushing these various side projects along and helping me with the bioinformatics. And I'm open for questions if you have any.
Fantastic. This is very exciting. I believe that Chris need to go for moving to new JCVI place. So I will start with some questions. This is a very exciting. Also, I have a lot of questions, but I will start with very naive question. So I actually I was excited to get DARPA grant many years ago, and then I actually have some issue, so I didn't pursue further. But I know a little bit, but not that much. But my very naive question is, you have any some bacteria that is already known to bind whatever, the rare earth element.
Hmm.
Can we do very simple experiment, uh, grow that bacteria a lot, and then pass through with individual rare earth element, including heavy,
Hmm.
as well, and then see before and after whether some amount kind of decrease. In that way, you could say, oh, this gonna be binding to whatever bacteria, protein or bacteria, and is utilizing those things. Can you do
Hmm.
those kind of simple experiment? I just wondering why we don't have some rare earth element not utilized. That might be because of toxicity, because you're showing that data,
Hmm.
some of the slide. But I just want to get some insight. Why is that? Why the other question is, I mean, another way I can ask the question is, over the whatever billions year of evolution, why we still couldn't find the bacteria utilizing some of the rare earth element? This is because of detection issue or toxicity issue?
Yeah, so we can kind of break that down into two parts. There's
Uh,
the why and the how.
You're right.
So the how, why is like this cutoff? Is it
Uh,
because, maybe they can take up all the metals, but they can only use some of them?
uh,
Or is there a selection filter up front where they can only import these rare earths?
uh,
And I think because it's biology, it's probably several layers of selection filters between import, utilization, storage, and sensing is probably a big one.
uh,
And that goes back to the why. In typical soil, the light rare earths are orders of magnitude more abundant than the heavies. So
uh,
evolutionarily, they're probably, incentivized to be specific towards the lights, and they never see the heavies, so there's no selection pressure to use them. Um, so
that
I
makes
think if
sense.
we want to find an organism that either preferentially or partially uses the heavies, we have to look in environments where there is more heavy elements. Um, and the big one it would be, hydrothermal vents. That's where you get areas where, say, holmium and dysprosium are not higher than the lights, but more in parity. So organisms there that use rare earths maybe can more broadly use them. And
That makes sense. So this is more likely because of the abundance in that specific area. The
yes,
because I mean, in terms of the function, this is more like a coordination role. It doesn't
yes.
matter, heavy or light from
It
the
does
bacteria
it
perspective
it does matter a little bit
a little bit.
because these get more electronegative as you go
Uh-huh.
left to right. So they
Right.
get more efficient at moving electrons around.
Uh-huh.
There's some work done on the Storkins methanol dehydrogenase and the paired cytochrome that it
Uh-huh.
uses to recycle the PQQ.
Uh-huh.
The irony there is it seems that heavier rare earths are too electronegative in that system, and they don't allow the electrons to leave the PQQ into the
Oh.
cytochrome. So the cytochromes that are paired with these methanol dehydrogenases that are lanthanide dependent
Uh-huh.
have an aberrantly low redox potential, so plus 160 millivolts, as opposed to the more standard 260 millivolts of other cytochrome Cs. Um, so the hypothesis there is if you raise the redox potential of the paired cytochrome, can you now efficiently move the electrons through the system despite the more, greedy heavy rare earths? And there's some evidence that that is the case. So Emic Storkins doesn't really like to use beyond samarium, whereas other strains like M. fumariolicum can use up to gadolinium. And that's because its cytochrome has a higher redox potential.
I
So I,
so
I think they haven't evolved to need to use the heavies, but
uh,
I think the way to get there is evolutionarily accessible. And from our evolution experiment thus far, we think that is true.
I see. So that means, I mean, we haven't found some organism utilizing that heavy one yet, but
Correct.
there
But I
might
think it's
be
I think
there.
something's out there that can definitely use at least a couple beyond here.
Yeah, that's kind of the kind of makes sense because the evolution doing for many many years,
Hmm.
the years we cannot even imagine. So I just wonder why we don't have those kind of system yet.
So
So
the
that makes sense.
one thing that have has kind of puzzled the field for the last 10 years is why there are lanthanide dependent enzymes at all, because the
Mm.
calcium one works just fine,
Mm.
and there's a lot more calcium in the environment than there is rare earths.
Hmm.
And there's a couple hypotheses. One, the calcium enzymes are less efficient because calcium is not as good of a Lewis acid as rare earths. So the
In
rare
terms of the
earths are more
strength.
efficient. Yeah, yeah, it's a +2 charge instead of a +3. So it's less
Mhm.
efficient at doing the chemistry.
Mm.
But also there could be the question of, well, calcium is used for a lot of different stuff. So maybe it's better to dedicate, lanthanides to this enzyme, let calcium be doing other things. Um,
Mm.
there's some evidence that the lanthanide enzymes are actually older and the calcium dependent versions evolved from them.
I see. That's
So
very interesting.
yeah, the lanthanide ones are much more widely distributed across taxa, and it seems like if you do these, evolutionary at trace analyses, the calcium ones kind of showed up later.
Hmm. I see. That's a very interesting. So, my second question is, actually, I was thinking about the separation using different
Hmm.
affinity, but you also showed the recent paper kind of done that, kind of
Hmm.
using the whatever protein, I guess. You kind of go through very quickly to, yeah, that paper. And
Yeah.
then showing the very good resolution in a sense. So, what is your opinion regarding this application? Is it possible to use this one for their commercial application in the future?
I think definitely
Not
yes.
large
So
scale?
I think this can scale up, and usually the question is always, can it scale up to the amount we would need it to? Which for especially a column-based approach, probably not. So you would have to do some batch process. But the kind of odd thing about rare earths is they're called the vitamins of industry because you don't need a lot of them. You just need them in everything.
Uh,
So the actual scale we would need to mine is magnitudes lower than iron or aluminum or copper. We don't
mm,
use nearly as much of these metals as we do these
mm,
others. So you don't need it to scale as well as you would need an iron process. So
mm.
I think this is definitely a path forward. The caveat for this experiment here is this was done with already soluble ions that you pass over a column. So you still need to mine, grind, and beneficiate the ore before you get to this step. This was also done with a pure solution of the mix. So we don't know how the system behaves if you have, say, huge differences in all of the rare earth abundances or competing ions like aluminum or calcium.
So that means, I mean, to use this system in a commercially viable way, you need to separate the iron or whatever the abundant, guys first, and then
Yeah.
do the rest of the separation.
And you can see from the structure of these proteins that it's just a pocket of really, really, really negative charge.
Mhm.
So it could presumably bind a lot of ions in these pockets.
Uh-huh.
It's kind of depending on the spacing between all the ions and whether everything coordinates just well enough to shield the charges.
Right.
So some tests on these proteins, especially from this paper, shows that it's more specific for rare earth than calcium.
Uh,
But we are getting a lot closer now than ever to having a bioprocess for
uh-huh.
purifying, which I think is huge. This is a really big deal paper. It's on bioarchive right now, so we're waiting for the publication to drop because they have structures of these proteins.
Uh-huh.
Um, but we still need something upstream if we want to go the full bioprocess route to leach the metals and maybe do some early enrichment, which I think is a good job for phosphate solubilizing microbes, um, which are kind of tailored to breaking apart these phosphate minerals. And
I
then you
see.
can pass whatever they don't use onto the column that has this protein, and then you have a full bioprocess that works.
I see. That's exciting. It based
Yeah.
on the all the names, it sounds like from the Chinese Institute.
Yes. It's
Uh,
from Tsinghua University in Beijing, I
okay,
believe.
I see. So the also they have the abundant rare earth element in that country. So they're
Hmm.
probably doing a lot of experiment at this moment because this is becoming important resources.
Yeah.
Okay.
And they also have, they're kind of unique in that they have these ion absorption clays,
Hmm.
which are higher abundance in the heavy rare earths, but lower overall grade.
Hmm.
So here in the US, the mines that produce a majority of the rare earth ores that we use
Hmm.
is Mountain Pass in California, which produces mostly bastnasite, which
Right.
is enriched for lanthanum and cerium,
Mm.
and less the heavies. So we actually have less ability to even get our hands on these heavier rare earth elements, other than from really low input grade materials like fly coal ash or e-waste. So enabling the use of those more sparse inputs is also going to be kind of important
I
for
see.
coming up with the demand.
Got it. The other simple question is how expensive to do this type of experiment? I'm just curious.
It's actually not that expensive
Okay.
because the rare earths we need only so little to do
So
these
literally.
growths. Yeah, so you only need a micromolar. So we use a couple milligrams every couple months,
I
which
see.
is fine. The buying the initial bottles of the salts, the scandium was really expensive. I think it was like $500 for 5 grams of it.
I see. I see. Good. My last question is, so the this current extraction process is very environmentally, the bad process.
Hmm.
That part I remember also you kind of briefly mentioned that one. The biology definitely have a lot of benefit.
Hmm.
But at the same time, I haven't done any, techno-economic analysis. And I believe you mentioned something, briefly, but have you done the techno-economic analysis, and then that kind of showing the benefit out of the
Yes.
compared to the chemical process we currently have?
Yeah, so you can be cost competitive with the typical. Let's just use the like high grade input as an example. You can be cost competitive with this type of process primarily because the energy you need to grow a lot of cells is a lot lower
Mm,
for getting a ton of metal, and that
mm,
depends on two major factors. One, the leaching efficiency. So if you have
mm,
a rock with 10 milligrams of rare earth, if you get all 10 out of the rock, then you're cost competitive pretty easily. The
mm,
other is if you're using a process where you're absorbing the ions into or onto the cell, there's a couple breakpoints you need to hit,
mhm,
and that depends on the metal. So for terbium, you need 0.25% dry cell weight if all of the metal is going into the cell to be
mm,
profitable, which is high but not unreasonable. To compare that to something like praseodymium, you need something like 4% dry cell weight, which is a lot more challenging of a target to hit.
mm.
Um, so it depends on the metal you're going for and the leaching efficiency primarily, but you can be cost competitive, assuming you can process the amount of material of low grade input that you would need.
Absolutely. That's fantastic. That's a very important resources, a lot of countries currently, seeking. So I hope, I mean, this your work kind of giving a lot of uh great insight and then potentially the commercialized, out of your process in the future.
What's really fun is all the groups that are working on this. Everyone has their own favorite flavor of trying to do this. So some are working with microbes, some are working with peptides, some are doing a
Mhm.
combination. Everyone's working with a different microbe.
Uh-huh.
So there's a lot of people working on this, and everyone has kind of a different idea of how to best go about it.
Sure. But we need, I mean, different approach,
Mhm.
to collectively successful because, it doesn't matter who, solve this problem as long as we have the multiple approach and then solve some portion of the problem. And that's how I believe, the science and technology moving forward. Not
Hmm.
necessarily one guy just doing everything. I mean, it's not happening nowadays because that is all collective and the
Hmm.
process. That's how
Yeah.
I uh, strongly believe, science and engineering should go.
Yeah. And I think personally, something like this is probably what the final process is going to look like,
Mm,
where you have some guys doing the, leaching and beneficiation, and then you
mm-hmm,
have probably proteins during the final purification.
mm,
Um, and that way you can do the whole thing without needing, the kerosene or the
mm-hmm,
TDP or any of the acids. Um, so way fewer pollutants being generated, more energy efficient,
absolutely.
and then, roughly cost competitive.
Right. I also call this type of thing is a hybrid process using
Hmm.
chemistry and biology or some mechanical and everything
Hmm.
together.
I think it's 20% of global copper comes from a hybrid biomining process.
Mm. So using some, biological process, probably later stage, I guess, right?
It's actually early. So
Only.
the way it works is PseudothioBacillus thiooxidans. They
Oh,
can directly oxidize inorganic sulfur and elemental sulfur. So you have these sulfide minerals like pyrite that has copper sulfide. It oxidizes the sulfide, breaks apart the rock. Also, incidentally,
I see.
yeah. So you end up with the microbes are catalyzing this self-reinforcing dissolution of the sulfide minerals, and then you can kind of just scoop everything off the top and purify the copper once it's in solution.
So that means this small bacteria is one of the superhero breaking
Yep.
up the rock.
And we've actually been doing, unbeknownst to us, biomining for a very long time.
Uh-huh.
There's a river that I believe it's in Spain that was red in color,
Uh-huh.
reddish in color, because the bacteria natively there were doing this biomining. And
Uh-huh.
so nearby residents would collect the water, precipitate the copper, and be able to get bronze from it.
I see.
So
That's interesting.
so we've actually been doing biomining a lot longer than we've known what biomining is.
Uh-huh. That's true. Yes. Fantastic. Okay, so now I see the 11:02 Central Time. That means your lunch time. So let me close and then we can start, still kind of chat a little bit more.
All right.
Okay. Thank you. And thank you all for joining and staying today. We'll meet again on November 13th, not the next week, but 2 weeks from now, but also different time at noon Central Time due to some conflict of schedule with the same Zoom link. We'll have the Professor Mark Guer at University Pompeu Fabra or Pompeu Fabra University, Spain, and also Dr. Justin Panisci at the Lawrence Berkeley National Lab. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. And thank you. And I stop recording. Just give me 1 second.